Hello students, I am Dr. Sachin Kapur and today we will be discussing digestion of proteins. There are two main sources of proteins in our diet, the animal proteins and the plant proteins. Animal proteins, they are called the first class proteins because they contain all essential amino acids in proper proportion. The plant proteins are second class proteins because they do not contain all essential amino acids in proper proportion. We'll discuss about essential amino acids in the topic of biochemistry. Let's start with protein digestion from buccal cavity. We know buccal cavity has saliva and saliva has salivary amylase, which is for digestion of carbohydrates, that is starch. As far as proteins are concerned, protein digestion doesn't happen in buccal cavity. So for buccal cavity, I have written here no protein digestion. After buccal cavity, the food goes into esophagus, and we know esophagus doesn't secrete any digestive enzyme. Next is stomach. Please remember that stomach is mainly designed for protein digestion. The wall of stomach has gastric glands, which are of different type: cardiac glands, fundic glands, pyloric glands. The gastric glands of the stomach, gastric glands secrete gastric juice, right? And this gastric juice has important enzymes which begin the protein digestion. A very important enzyme in the gastric juice is pepsin. Pepsin is secreted in an inactive form, that is pepsinogen. This pepsinogen is converted into active pepsin by hydrochloric acid. Once a small amount of pepsin is formed, it can convert the remaining pepsinogen into pepsin, that is autocatalysis. So pepsin shows autocatalysis, means when few molecules of the active pepsin are formed, they can convert the remaining pepsinogen into pepsin. Now, which cells of the gastric glands, they secrete pepsinogen? It is secreted by chief cells, which are also called zymogen or the peptic cells. The chief cells of the gastric glands, we'll discuss about gastric glands in detail in the structural anatomy of the stomach, right? So the chief cells, they secrete pepsinogen, which is activated into pepsin by hydrochloric acid. HCl is secreted by oxantic cells. HCl is secreted by oxantic cells, which are also called parietal cells. When the pepsin is activated, this active pepsin starts protein digestion. It breaks proteins into proteoses, peptones, and large polypeptides. Pepsin works at an acidic pH of 2 to 3. If the pH is beyond 5, pepsin is inactivated. So the acidic pH is created by hydrochloric acid, right? Pepsin begins protein digestion by converting proteins into proteoses, peptones, and large polypeptides. There is another enzyme in the stomach for protein digestion, that is renin. Renin is also secreted in an inactive form, prorenin. Prorenin is converted into renin by hydrochloric acid. Renin acts on milk protein casein. The milk protein casein is acted upon by renin to form paracasein, which in presence of calcium ions forms calcium paracasinate. I'll repeat, renin, which is present in the gastric juice of mammals only, because renin is related to digestion of milk proteins and milk is produced by mammals for their young ones. So this renin is present in gastric juice of mammals during infantile stages, because milk is a diet for infants. With the advancing age, the production of renin reduces. In humans, renin is also called chymosin. Renin is also called chymosin. So it converts casein into paracasein, which in presence of calcium ions forms calcium paracasinate. This calcium paracasinate is acted upon by pepsin to again form these proteoses, peptones, and large polypeptides. I have written the spelling here. So calcium paracasinate is broken into proteoses, peptones, and large polypeptides. That is what is happening in stomach. I'll repeat. Stomach is chiefly designed for protein digestion. The enzyme, most important enzyme, is pepsin. Pepsin is absent in case of invertebrates. Pepsin is a very strong enzyme and it can even digest collagen. When the bolus of food gets mixed up with the acidic contents of stomach, it is called chyme. So all these things are happening in the chyme. Next we move to small intestine. Let's see how the protein digestion continues in small intestine. We know that small intestine receives 3 digestive juices. Number 1, bile juice. Bile juice doesn't contain any digestive enzyme. So for bile juice, I am writing no digestive enzyme. Next is pancreatic juice. Pancreatic juice is the richest digestive juice. It has variety of enzymes for digesting different types of food components. Pancreatic juice has enzymes like trypsinogen, chymotrypsinogen, procarboxypeptidase. Trypsinogen is inactive, so it has to be first activated into trypsin. Similarly, chymotrypsinogen is also inactive. It has to be activated into chymotrypsin. Third is procarboxypeptidase. This is activated into carboxypeptidase. Trypsinogen inactive, chymotrypsinogen inactive, procarboxypeptidase inactive, and these are the active forms. Trypsinogen is converted into trypsin by enterokinase, or it is also called enteropeptidase, which is secreted by duodenum when the pancreatic juice is released into duodenum, which is a part of small intestine, right? So that activates trypsinogen into trypsin. Once the trypsin gets activated, it converts chymotrypsinogen into chymotrypsin, and procarboxypeptidase is converted into carboxypeptidase by trypsin. When these 3 enzymes they get active, they act on proteoses, peptones, and large polypeptides. These 3 enzymes, trypsinogen, chymotrypsinogen, procarboxypeptidase, they act on what? These act on proteoses, peptones, and large polypeptides to form what? To form small polypeptides, dipeptides, and few free amino acids. Proteins are polymers of amino acids. Ultimately, the protein digestion will be completed when the proteins are broken down into amino acids. But here still we are left with small polypeptides and dipeptides. These also need further digestion. So the intestinal juice produces has, I'm writing the enzymes, amino peptidases and dipeptidases. aminopeptidases break small polypeptides into amino acids, right? Dipeptidases act on what? Dipeptides to form amino acids, right? So the intestinal juice, which has aminopeptidases and dipeptidases, has finally completed the protein digestion. So we can say that the end products of protein digestion are what? Amino acids. End products of protein digestion are amino acids. So these amino acids are absorbed easily through villi of small intestine, right? Protein digestion, let's have a quick recap. Proteins of animal origin or of plant origin, as far as buccal cavity is concerned, there is no protein digestion. Esophagus, no protein digestion. Stomach, protein digestion begins. Most important enzyme, pepsin, secreted in inactive form, pepsinogen, converted into pepsin by hydrochloric acid. Pepsin starts protein digestion by breaking proteins into proteoses, peptones and large polypeptides. Another important enzyme in the gastric juice is renin. This renin is in the gastric juice of mammals only. Renin converts the milk protein casein into paracasein, which in presence of calcium ions forms calcium paracasinate, and that is acted upon by pepsin. When the chyme enters into the small intestine, it receives three digestive juices. Bile juice, produced by liver, stored in gallbladder, do not contain any digestive enzyme. Pancreatic juice contains important protein digesting enzymes in inactive form, because if they are activated in the pancreas, they will digest the pancreas itself. Trypsinogen converted into trypsin by enterokinase secreted by duodenum. Trypsin is also called ancestral enzyme because it is found in all the organisms starting from protozoa to primates. Chymotrypsinogen converted into chymotrypsin by trypsin. Procarboxypeptidase converted into carboxypeptidase by trypsin. When these three enzymes get active, they act on proteoses, peptones and large polypeptides to form small polypeptides, dipeptides and few free amino acids. Finally, the intestinal juice has aminopeptidases and dipeptidases, which will break these small polypeptides and dipeptides respectively to form amino acids. So we have seen that the end products of protein digestion are amino acids, which are absorbed easily through the villi of the intestine. So protein digestion started in stomach, completed in small intestine. So with that, we end up with this lecture here only.